cancer section. And then we've got a section that covers the oncogenes and a section that covers the tumor suppressor proteins. And that's basically what tumors are. Tumors are a balance of oncogenes and tumor suppressor proteins. And how that balancing out plays out is going to be what determines if a cell becomes tumorigenic or not. All right, so additionally, it's no single mutation is significant enough to cause cancer. Cells don't work like that. Cancer, what we think about cancer, how we think about cancer, we think about end stages of tumor development in which the tumor has picked up many, many mistakes over time, and those mistakes have given it a very big edge in proliferating out of control, in avoiding death, in not changing, not differentiating, and in other things as well. How it interacts with the extracellular matrix changes. All these things are what make tumors tumors. All right, so just some important terminology here. Neoplasms are tumors. Benign tumors are localized and non-invasive, right? Development of a benign tumor, no matter where it is not necessarily the end of the world. Any quack surgeon can go in and take out a tumor, right? If it's benign, it's not going to spread. The problem is when it spreads. When it spreads and you have millions of tumors now growing all over your body, no quack surgeon can go in and pull that out. So that causes problems. So metastasis is a very important component of cancer development that makes cancer very deadly and dangerous. All right, types of cancers include the carcinomas, which arise from epithelial cells. Most cancers are carcinomas. Sarcomas, which derive from connective tissue, are less common. You have leukemias, which are from the leukocytes. So the leukemias, though, exist and it's confusing between leukemia and lymphoma. Leukemias exist in the bone marrow, right? So they're the cells that exist in the bone marrow that don't differentiate. So you get, like T cells that don't form mature T cells. They remain in that like plasmodial immature state and then divide out of control and circulate, right? So you don't get tumor formation in the bone marrow, but it's the cells of the bone marrow that are becoming leukemic cells. Whereas lymphomas do form solid tumors, and they typically form in the bone marrow, also the lymphatic tissue. Right? Benign tumors are localized. Malignant tumors undergo metastasis. Stages of cancer, they all do start with a single mutation. And that mutation gives the cell some sort of proliferative advantage. It starts to divide heavily. And as it divides heavily, the problem with heavy division is now the chances of acquiring new mutations are increased. So the likelihood of it acquiring additional mutations increases just from that initial mutation. So you start to get some abnormal development of a mass, and through subsequent rounds of proliferation and cell cycle, you start to select the cells that are dividing more rapidly. They also have survival advantages because cell growth is not the only thing that is activated in cancer. An inhibition of apoptosis is also activated. All right, good example is in colon cancer, how the adenoma becomes a carcinoma. All right, you can see up here, you've got your basal lamina, you got your connective tissue under it, you've got your lymphatic vessels and your circulatory vessels, and you get one single cell that acquires a mutation and it starts to divide, and those cells accumulate more mutations. And now you have an adenoma, right? And adenomas are what people go into colonoscopies hoping to have removed, and they don't have something that's gotten more progressed than that. Eventually, it's going to develop more and get bigger into an intermediate adenoma, and later on, it will become a carcinoma. So what's forming it into a carcinoma is invasion into another tissue level. Remember, carcinomas are tumors of the epithelium, right? So this tissue in here. And then pieces of the tumor, the cells of it can break off. They can invade further. They can get into the circulatory system, and then that's bad after that because it's spread. Wherever the tumor lodges in the body, it's going to develop into a new tumor. And this is the process of metastasis. So, and then a lot more things are happening in this cellular process. So under normal conditions, like when you grow cells in culture, and if you grow just like typical normal fibroblasts that haven't been immortalized, they will undergo what's called density-dependent inhibition, which means when they start getting to a certain density, they will enter a quiescent stage, and that environment becomes very toxic because they'll undergo apoptosis. All right, when they start getting crowded like that, they want to die. Tumor cells, though, when they start growing and become very high density, they don't have that problem. They'll continue to grow even after that. They'll grow on top of each other, and they won't undergo apoptosis. So there are obviously contact cues through integrins that mediate this property where you have this inhibition of high density and you get unrestricted proliferation. So in the top there, you've got like normal fibroblasts. Here you've got some, that looks like a HepG2 cell line, it's a liver cell line. And see, normally they grow. You can see they're starting to look sick up there. Like the membranes are starting to bleb and stuff, and that's apoptosis starting to occur. But they're inhibited. They just grow in a monolayer. They don't grow on top of each other. Cells that are tumorigenic, though, will look healthy and also start to grow on top of each other. Yeah.
How does this relate to, like, you know, if you get like one random hair on your arm that grows super long? Do you have any idea what I'm talking about or no? It's like why is it not regulated? Because like all the other hairs on my arm,
Yeah.
like, you know, stop at a certain length.
I don't think that's the tumor. I don't think, but I know what you're talking about, man.
I'm just trying to see that like kind of relation.
I had this professor, he would get this like long nose hair that would grow out. And I always begged him, just pluck the thing out, man. I don't know what causes that. It could just be I don't know what causes that. Maybe it's that you got to change your aftershave or something. I don't know. I don't know. But it's not, that's definitely not tumor, like that whole process of hair follicle growth, which I'm not expert in. I think has other cues that get that going. So interesting question. All right. So in culture, you can see in with tumor cells, they'll pile on top of each other and start to grow like that. Yeah.
Might be a stupid,
There's no stupid questions. What up?
but like, is any cell proliferation cancer technically?
No, I would say no.
Like a wart, for example. Like if you
But
have-
a lot of those things are precancerous, we say. And just like the adenoma, that's a precancerous condition because you're already on your way. Given enough time and the right conditions, yeah, it will form into it. A lot of warts, though, are viral based, right?
Yeah.
And viruses are heavily linked to cancer. We're going to see that in this chapter. That's a good question. All right, so what are cancer cells? What do they do? Well, they don't change. They don't die. Through many, many rounds of replication, because of the limitations of the Okazaki fragments, right? And the lagging strand, the chromosomes should be shortening every time but tumor cells have found a way to get around that and keep their telomeres at a consistent length. Most tumors have hyperactivated telomerase to overcome that. Autocrine growth stimulation, tumor cells will secrete growth factors that work on themselves to keep those pathways going to maximize the proliferation. They will react differently to their microenvironment. They won't be as adhesive, and that increases their level of metastasis. It also maximizes their cell signaling to increase their proliferation and reduce their apoptosis, because cell adhesion and how cells interact with the microenvironment is important through integrin signaling and whatnot. Invasion of surrounding tissue. So tumors will release proteolytic enzymes to degrade connecting proteins like collagen and fibronectin, break them down so that it can get through tissues. And then angiogenesis, all right? Even at a very small size, tumor requires some level of angiogenesis. It requires the development of new microcapillaries in the tumor to provide oxygen and nutrients for the growing tumor. This is a heavily researched area of cancer biology that hasn't really resulted in too much treatment, but it's not given up on, because really, if you can, if you inhibit angiogenesis in the body, that's one way to really effectively treat tumors and not have a lot of side effects. Because the thing with cells, or thing with cancer cells, is there are cells, right? So a lot of these ways to treat cancer cells are going to produce heavy side effects. All right. So I just went over all these. This is talking about autocrine. It's autocrine pathways, release something that works on a receptor that's on the cell. Yeah, these autocrine stuff, you can see the cancer cells releasing those little balls, right? The balls activate receptors on itself. That tells the cell to divide, go through the cell cycle. Block differentiation. The three main hallmarks of cancer are they grow out of control, so increased proliferation, they don't die, decrease apoptosis, and they don't change. And this is especially true in the blood cancers. They stay in this early primitive state, which is an early step in hematopoiesis. You don't get activated T cells and activated B cells. So people that have these leukemias and lymphomas usually have very poor immune systems to go along with their cancer, because they don't have those mature B and T cells. All right, big step in cancer is evasion of apoptosis. These cells that are dividing out of control should kill themselves, and they don't. And also the maintenance of the chromosome ends. I think it's more than 80% of tumors have a hyperactive or constitutively active telomerase enzyme, which replaces the DNA at the ends of the chromosomes to kind of make that stable. All right. What causes cancer? Well, carcinogens, right? So a lot of these carcinogens will add bulky groups to the DNA. DNA damage, right? So they can damage DNA by adding bulky groups to it. You can also get DNA damage through ionizing radiation and UV radiation, right? So also tumor promoters, right? And what do we mean by this? Not like a promoter on the DNA that's driving expression. What we mean is, like, over time, as the tumor is progressing, things that kind of occur in the body that help the tumor develop. Inflammation and the signals that are associated with inflammation are something that can do that. Cytokine release and interleukin release from immune cells will work on tumors and kind of promote them. So it's like your body's immune system. Not only does it not get rid of the tumors, but it's actually helping them grow. And then viruses. So viruses are intricately linked to tumor biology. They can insert basically new genes into the genome that cause it to proliferate out of control. They can disrupt genes that are in the genome that are required to kind of suppress or put the brakes on cancer. And a lot of different families of viruses can induce cancer. There's a table in here, that's right. So DNA viruses and RNA viruses can both kind of induce cancer. You can see these are just some common ones here. Hep B can cause liver cancer. Hep C, also liver cancer. Herpes virus, papilloma virus, all these guys, when they infect, they can present new oncogenes to the cell, or they can inhibit tumor suppressor proteins. Yeah.
Yeah.
So you mean like using viruses to treat cancer? Yeah, so you want to talk about that? Didn't you do a presentation on that a few weeks ago?
So, they actually use retroviruses to, in, terms of T cells, they can insert genes into the T cells to make, antigen receptors, they're called chimeric antigen receptors. And they can latch onto tumors and disperse them. So, yes, there's other ones too, that aren't retroviruses, but those are usually the most.
What are those called again?
With our T cells.
What are they? Yeah, CAR T cells,
Yeah,
right?
CAR T cells.
Yeah. So, yeah, like, and that's the most significant advances in oncology have been through manipulating the immune system. So taking cells out of a person, changing them a little bit, growing them up, and then introducing them back into the person to fight the cancer. That's been, really the most successful way and safest way to battle cancer. Yeah, that uses viral mechanisms to do that.
So the
So
process called seeding.
what is it?
Is that process where you take out and then put back in called seeding? Or what's that process called?
CAR T immunobiology is what it is. But yeah, so whenever you're seeding cells, you're introducing them into a new vessel. So yeah, if you're giving them to a patient, you are seeding them with
It's
cells.
also radiation oncologist. I've heard like you're talking about doing seeding procedures.
Yeah. Yeah.
That's kind of what you're talking
Yeah,
about.
I think that would be it. The thing with all this immune biology and treatment is it's really, really expensive. It's really slow. You have to grow the cells up, it's like personalized medicine, so it hasn't really taken a hold yet, but um, yeah, that's true. And that's a big problem with cancer. Most people, they go in getting treated for multiple myeloma and they walk out the next week having leukemia too. Because the drugs that are used to treat cancer, the chemotherapy drugs, one of the side effects of them is that they cause cancer because they damage DNA and so yeah, it's a very a very tricky disease to treat. All right, so what tumors are though, what they're caused from initially, like that single mutation and those mutations that the tumors pick up over time, it's basically one of two possibilities, right? Either a gene called a proto-oncogene gets activated, mutated into an oncogene, or another gene called a tumor suppressor gene gets shut off. Those are your two possibilities. The cell has many, many proto-oncogenes in it, and those are genes that are normal genes that we need and the cell needs to survive, but when they're mutated, they become dangerous. And think about it, they are going to be genes that are involved in cell cycle progression, right? If they get hyperactivated, now the cell's going to go over, divide out of control. They are genes that oncogenes, another class of oncogenes would be genes that are anti-apoptotic, right? So if they're anti-apoptotic and they get mutated, hyperactivated, the cell won't die. The tumor suppressor proteins are opposite. Okay, and we'll cover that there in a little bit. So this section overviews the oncogenes. They were discovered early on from viral infections. There's particularly RSV, so not the respiratory infection. RSV stands for Rous sarcoma virus, which is a virus that's common in chickens. Viruses are not required to cause cancer, but many of them do, because they can introduce new genes or oncogenes. Right? Good example is this RSV. So they had two different viruses that they looked at, Ralph sarcoma virus and avian leukosis virus. And they looked at the effects that they had on chicken embryos. And you can see RSV transformed the cell, but ALV did not, right? So some virus can induce tumorigenesis, some viruses can, some viruses don't. The reason RSV could is because it was inserting an oncogene into the target cell called VSARC, which stands for viral SARC. And SARC is a signaling protein that we talked about in our signaling chapter. Yeah.
Do some viruses intentionally cause tumors in order to proliferate themselves faster? Or is it-
I don't know if it's an intentional thing. Like you think about even in terms of evolution, it's better for the virus.
Yeah.
I don't know about that. I don't think that is a deciding factor because really you ended up killing the organism. But the way that viruses function enable them to do this, right? So retroviruses, especially retroviruses, use reverse transcriptase, right? They also incorporate in the genome of the cell. So when they incorporate in the genome and then they're reverse transcribed out, now they're going to pull new genes out from the target host gene. Those can become, if they mutate in that highly mutagenic reverse transcriptase, you can get a proto-oncogene forming into an oncogene. Okay, so some viruses have it. RSV has this SARC tyrosine kinase. When you look at viruses, they have pretty simple structures. They have three basic gene groups. They have the GAG genes, which are important for the structural proteins, also the viral protease. The POL, which stands for polymerase, codes for any reverse transcriptase, and also the polymerases that are needed to make the viruses. And then the ENV genes, which code for the protein, glycoprotein coat that makes up the outside of the virus. Okay, so these are the genes that are required for all viruses in order for them to hijack the machinery and reproduce, but additional genes can get added in, right? For example, if you compare the ALV versus the RSV sequence, you see the GAG-POL envelope genes in all three of them, but that RSV has incorporated that SARC protein. Where did that SARC protein come from? Where did it get it? I mean, not originally. Originally, because RSV probably derived from ALV. So ALV picked up SARC and became RSV. If you look at viral evolution here, that's probably what you're looking at. Where did it get that SARC gene from? So the chicken, right? So it probably got it from chickens that it was infecting, because SARC is a normal cellular protein, right? It's involved in cell signaling, JAK-STAT pathways, and it's picked it up through its incorporation in the genome. Right? The viral DNA incorporates in, and then it gets pulled out through reverse transcriptase. Yeah.
Is there a limit to how much, like, virus?
Well, the more genes it gets, the more unstable it's going to get, right? These things are always trying to stay as small as possible. But I mean, we use retroviruses in the lab to transfer and transmit a lot or induct a lot of different size genes. So it's got, it doesn't have too much of a limit there, but I'm sure that's been quantified. I just don't know the answer. Yeah, that sounds like something they would have needed to figure out because these viruses are used as induction agents for introducing new genes into cells. All right, so this SARC gene at one time, was a proto-oncogene, but here it became mutated as a version of VSARC, and now we call it an oncogene because it can induce cancer. All right, what are some other proto-oncogenes. So VRAF is a good example. All right, VRAF talked about, I think I have the figure, or I don't. There's RAF, that would be RAF right there. So VRAF proto-oncogene, RAF has a regulatory component and then it has the kinase component. And if you get rid of the regulatory component, like through viral incorporation, now the kinase is constitutively active and that whole AKT pathway gets permanently turned on, and that drives proliferation of the cell. Okay, so as a proto-oncogene form, it's got something suppressing the kinase component, but in the oncogenic form, you've gotten rid of the regulatory protein, and there we just have some generic viral protein, like some GAG protein that's been put into it. Okay, other oncogenes. RAS is a big oncogene, very frequently mutated in human tumors. There's like 3 different isoforms that have been identified: HRAS, KRAS, and NRAS. And just specific point mutations, I think, is that the same mutation we saw today in the exercise? We're talking about RAS. I think it was that G12V. Is a different one? Oh, no, okay. So particular point mutations can make them hyperactivated. So RAS, is a G protein. And if you keep it locked in that GTP state, then that pathway becomes constitutively active. And if that's a pathway, which RAS pathways are, which are involved in cell growth and division, well, then you have a problem. Right? Normal RAS picks up a mutation, it becomes HRAS. Right? Here's RAS right here. It's got a parental group that allows it to stick inside that thing. It keeps it prenylated and associating with the membrane. So it gets activated by GEF. There's the SH2 domain that's interacting with the RTK. We talked about that. And RAF will activate GTP there, activate MEK, and then ERK, and that whole kinase cascade. Okay. And I can barely see that figure on the left there. What is that? You got bladder cells, DNA, oncogene, detection of human tumor oncogene by gene transfer. Okay. Looks good. Okay, what else? Chromosomal translocation. So these are like moving big pieces of the chromosome around the genome. These can be responsible for many different types of oncogenic development. So c-MYC is a common one that's activated in Burkitt's lymphoma, and it moves from chromosome 8 to chromosome 14. So what is the implication of that? Well, so when you move this from here to here on chromosome 14, it's part of the whole immunoglobulin production mechanism. So, think about it. When antibodies are produced, they're produced at a really high rate. So you obviously have some really strong promoters there at the immunoglobulin locus of that chromosome. So moving c-MYC over there, now you're going to get a lot of transcription, translation of that c-MYC protein. c-MYC is a transcription factor, right? And results in increased levels or increased activity of AKT pathway, RAS MAP kinase pathway, levels of cyclin D and E. All these things are driving cancer. Another common chromosomal translocation is BCR-ABL. This is a very specific type of mutation that produces something called the Philadelphia chromosome, which is like a really short little chromosome. And it's common in CML, chronic myelogenous leukemia. Interestingly, with this specific mutation, there's a really good drug that treats this mutation called Gleevec. Like 100% remission rates. The problem is patients eventually get resistant to the drug. But why it's so effective is because it specifically inhibits the proteins that are made from BCR-ABL. So what happens is ABL protein moves over to chromosome 22. So now you have this fusion protein of BCR and ABL, and that makes the RTK constitutively active. So what else? Gene amplification, ERB2. This one's not that important. Okay, this one's very important. Functions of oncogene products. Right, so the RAS MAP kinase pathway, this is like a very central pathway in tumor development. Constitutively active RAS, you get a constitutively active pathway all along here, right? So here we've got RTK that's becoming, this is constitutively active. So you don't need like a growth factor to autodimerize it. It's always autophosphorylated. So it's always keeping that pathway on, okay? So, you know, these larger pathways, there's a lot more different points that can be affected. And that's what this figure is showing. It's, you know, you can have mutation at the level of the receptor. You can have mutation at the level of the intracellular signaling molecules, or even the transcription factors themselves. So mutation at any of these levels are potential oncogenes, since all of these proteins in this pathway work to drive expression. There's also no inhibitory steps in this pathway. If there was an inhibitory step, that would be not an oncogene, that would be a tumor suppressor protein. Okay, so growth factor receptors, you know, like we talked about the PDGF receptor, right? So in order for the PDGF, which is an RTK, in order for that receptor to get activated, a ligand has to bind to it, right? But if it's mutated to become constitutively active, then it doesn't need a ligand to bind. So here's normal RTK activation. PDGF binds the receptor, it dimerizes, it autophosphorylates. But in mutated PDGR, it's always stuck together. It's always self-dimerized, and it's always phosphorylated. So the cytoplasmic side of the receptor is always on. Its kinase activity is always strong. So there's a specific type of mutation that's allowing this to dimerize. You get replacement of the extracellular side with this sequence from the specific transcription factor, and that causes it to dimerize. All right, the last oncogene we're going to talk about is AP1. So here you've got two end products of the RAS MAP kinase ERK pathway, right? So AP1 as a transcription factor, right, is formed by the dimerization of Phos and Jun, and it drives expression of cyclin D1, right? What is cyclin D important for? It's important for that G1S transition, for getting past the restriction point. This is an example of things that are affected in the nucleus, transcription factors being affected, okay? Again, these long pathways, especially the ones with the amplification, MAP kinase, all those proteins represent opportunities for mutation and possible oncogenes. Okay, whoops. All right, last slide here. Okay, so now the thing with apoptosis, the proteins involved in apoptosis, it depends on the protein, but it can be either oncogenic or tumor suppressor, right? So if something is pro-apoptotic, right, what do you think? Is that a potential oncogene, or is it a potential tumor suppressor protein? Right. If it's pro-apoptotic, it's tumor suppressor because the tumor, we want the tumor to die, right? So a tumor suppressor. If it's anti-apoptotic, like the BCL2 proteins, that's potential oncogene. All right, so we've talked about this pathway here. Let's just review AKT. It inhibits BAD and FOXO, inhibition of BCL2, right? So the potential oncogenes here are all like with the glowing stuff. And these represent different tumor suppressor proteins. All right. What time is it? 11:50? It's getting a little. I think what we're going to do is not go through the rest of these guys just for the sake of time. And we'll wrap up tumor suppressor genes and then be done with it. Okay? Does that sound good? Getting a little long here. I'm never scheduling this class like this again. This is crazy. Okay, so tumor suppressor genes are the opposite of oncogenes, right? Tumor suppressor genes are trying to stop cell cycle division, right? They're trying to kill the cell. They're pro-apoptotic. Okay. So the big difference between tumor suppressor genes and oncogenes, though, is in order for the mutation to carry through, with oncogenes, you only need to have one allele mutated. But with tumor suppressor genes, what we found is both alleles need to be mutated. That's what it's called, the two-hit model or two-hit hypothesis. With tumor suppressor genes, both alleles need to be mutated. So, if you think about it, it's more work for the cell to knock out a tumor suppressor gene, right, than it is necessary to activate an oncogene. Because you need to get two alleles knocked out. So, if one allele is intact, it makes enough protein, that you get the function of a tumor suppressor gene. And we know this through these fusion studies. You've taken normal cells and tumor cells and you've fused them together. You kind of lose that tumor cell ability. It's the resulting fused cell is not a tumor cell. And that's because you've provided a functional allele from the normal cell. And this has later been described as the two-hit hypothesis. So for tumor suppressor proteins, you have to knock out both alleles. All right, this is true in the RB protein. It stands for retinoblastoma. So individuals with RB mutations, they develop these tumors in their eye. And heredity has a lot to do with it because if a child inherits one inactive RB allele, then all it needs to do is get the other allele inactivated and it has full-blown retinoblastoma. But people who do not inherit one of the mutated alleles, both alleles have to be undergo what somatic mutation to for the person to get cancer. Kids that are born with one inactive RB, they can still get tumors. It's not as proliferative if they have two, not as deadly. All right, what is RB? Well, we talked about RB in cell cycle. RB regulates cell cycle by inhibiting E2F, right? So RB binds to E2F. That represses transcription, but CDK4/6 and cyclin D release RB from E2F, and E2F is the transcription factor, right? That drives transcription of the other cyclin-dependent kinases and cyclins. All right, so if you inherit one defective RB, then you only have one more allele that needs to be mutated, and you can get tumor development. If you don't inherit a mutated RB, your chances of getting RB is very, very unlikely because you have to undergo two somatic mutations at RB in order to get tumor development. All right, PTEN is also a very highly implicated tumor suppressor protein. P10 works in the PI3 kinase AKT pathway. So if you remember, PI3 kinase is a SH2 domain containing protein that binds to growth factor receptors once they've been activated. PI3 kinase then converts PIP2 into PIP3. That's the inositol sugar that's integrated in the cytosolic side of the membrane. PIP reactivation activates AKT, right? We know what happens after that. So there's a protein called P10, the tumor suppressor protein, that can convert PIP3 back into PIP2. So it's antagonistic of this pathway. Whoa. How did I do that? Uh-oh. I don't know how to get out of that. I just touched the screen and it did that. Okay, so the AKT PI3 kinase pathway, that's a growth pathway. It's like an oncogenic pathway. So here's a protein that's involved in it that we haven't talked about yet, P10, which is antagonistic of that pathway. So it's slowing down cell cycle growth. So it's thus a tumor suppressor gene. Right, there's P10 up there, antagonistic of PI3 kinase. All right, P16. Talks about P16 in the cell cycle chapter. P16 antagonizes the CDK4, 6, and cyclin D complex. Right? It does that by keeping RB active. So if RB is active, it's bound to E2F and it's repressing, the transcription of genes that initiate cell cycle. However, when P16 is mutated, you get that inhibition removed and RB is not bound to E2F and it promotes uncontrolled proliferation, right? So this is going back, I should have kind of included here this figure, just to remind you, and this one. So P16 inhibits the phosphorylation of RB, right? Phosphorylation of RB is what releases RB from E2F. It prevents E2F binding. If it's not bound to E2F, then E2F is driving transcription of genes that keep the cell cycle going. All right, so P16 is a prominent tumor suppressor gene. All right, and finally P53. P53, the guardian of the genome, a transcription factor, right, that can be phosphorylated, that accumulates in the cell over time, but is readily gotten rid of, right? So P53 gets tagged for destruction by the proteasome through ubiquination, right? The protein that is tagging it is called MDM2. So MDM2 polyubiquinates P53, so it gets degraded. But if P53 is mutated, it doesn't interact with MDM2, right? It actually can be like phosphorylated to block, and you get a big accumulation of P53 in the cell. What does that do? Well, it causes cell cycle arrest. Okay. It also affects apoptosis as well. So P53 affects both pathways, right? Apoptosis and cell cycle arrest. We saw that previously. All right, last thing here is BRCA. BRCA1s are BRCA1 and BRCA2, very prevalent in breast cancer. These are DNA repair enzymes. Right? So if it's repairing DNA, that's